Hello and welcome to another Focus Deep Dive. Today we're embarking on a really crucial journey. It's through a foundational topic for class 11th and 12th board exams, and especially, for NEET aspirants, breathing and exchange of gases. I've come prepared with my questions, definitely eager to understand this vital process thoroughly. Our aim today is basically to simplify, clarify, make sure you get a robust understanding of this chapter, making these complex biological processes crystal clear for your exam readiness.
Indeed. And hello! This deep dive into Chapter 14, Breathing and Exchange of Gases, it's absolutely central to human physiology. It's not just about, what happens. It's really about why it happens and how it's meticulously regulated, all of which are critical concepts for your competitive exams. We'll be breaking down how our bodies manage this continuous life-sustaining exchange. We'll cover every essential detail from your NCERT textbook, which is, absolutely crucial for both board exams and NEET.
All right, let's begin by addressing the very basics then. Why do organisms, including us humans, need oxygen so desperately? And why is carbon dioxide such a concern that it needs to be removed? It seems fundamental, I know, but what's the core biological reason for this constant exchange?
That's an excellent starting point. It really underpins everything else we'll discuss today. So at its core, oxygen, or O2, is absolutely essential. Why? Because organisms utilize it indirectly to break down simpler food molecules. Think of substances like glucose, amino acids, fatty acids. These are your body's main fuel sources, right? When these molecules get broken down through various metabolic pathways, energy is derived. This energy, primarily in the form of ATP, is like the direct currency that powers every single cellular activity in your body, from the slightest muscle twitch to, complex thought processes, nerve impulses, even just maintaining body temperature. And it's an indirect breakdown because oxygen doesn't directly, chop up the chemical bonds. Instead, it acts as the final electron acceptor in the long chain of reactions, the electron transport chain that ultimately releases the stored energy. Without oxygen, these really high-efficiency energy-producing pathways, they just can't proceed.
Okay, so oxygen is all about generating that essential energy, the ATP. And carbon dioxide, what's its story?
Exactly. So while oxygen is consumed for this critical energy production, carbon dioxide, CO2, is simultaneously produced. It's a byproduct of these very same energy-releasing reactions, what biologists call catabolic reactions. Now, unlike oxygen, CO2 is considered a harmful waste product. If it starts to accumulate within the cells and tissues, it can significantly alter the body's pH balance. It makes the internal environment more acidic. And this pH change, it can then disrupt the intricate three-dimensional structures of vital enzymes, interfering with their activity and generally just impairing normal cellular function. So it's not just about continuously supplying oxygen to keep those energy processes running. It's equally important that the CO2 produced by those cells is continuously and efficiently released out of the body. This constant in and out flow, the continuous supply of O2 and removal of CO2, that is the fundamental essence of breathing.
I see. So breathing is that visible physical movement, like when we actually feel our chest moving up and down. But I remember learning about cellular respiration, I think it was back in chapter 12. Are breathing and cellular respiration the same thing, or are they distinct? This point often confuses us students,
That's a crucial distinction, and yes, it is indeed a common area of confusion for students preparing for exams like NEET. Let's clarify this thoroughly. Breathing, which is often referred to as pulmonary ventilation or maybe external respiration, is precisely what you described. It's the physical process of exchanging O2 from the atmosphere with CO2 from the cells. It's the macroscopic act of inhaling and exhaling, involving your respiratory muscles and lungs. You can consciously control it to some extent, right? And you can certainly feel your chest moving with each breath. That's the physical manifestation of this vital exchange. Cellular respiration, on the other hand, is a completely different process. It's a biochemical, a metabolic process that happens inside your cells, specifically within the mitochondria. This is where the O2 you've taken in is actually utilized by the cells to fully oxidize food molecules like glucose to release the maximum amount of energy, the ATP. It's a complex series of chemical reactions. Think glycolysis, the Krebs cycle, the electron transport chain, which you would have studied in detail elsewhere. So while breathing facilitates cellular respiration by supplying the necessary oxygen and removing the waste carbon dioxide, they are very distinct processes. One is a physical act of gas exchange at the organism level. The other is a chemical process of energy production at the cellular level. Understanding this difference is absolutely key.
Okay, that makes perfect sense now. Breathing basically sets the stage for cellular respiration to happen inside the cells. But do all animals breathe in the same way? Or do they have different organs for this? maybe depending on their environment, like water versus land, or how complex their bodies are?
That's an excellent question, and it really highlights the incredible evolutionary adaptability across the animal kingdom. You're right, the mechanisms of breathing vary quite significantly among different groups of animals. It primarily depends on two main factors: their specific habitats, whether they live in water or on land, and their overall levels of body organization or complexity. This diversity really showcases how evolution has tailored specific and efficient solutions for gas exchange to meet unique environmental demands. Let's break this down systematically, as this kind of comparison can be a common type of question in your exams. For the simplest forms of life, often called lower invertebrates, think sponges, coelenterate like jellyfish and hydra, and flatworms, you won't find any specialized respiratory organs. Instead, they manage their gas exchange, taking in O2 and releasing CO2, by simple diffusion directly over their entire body surface. This works for them because these organisms typically have a large surface area compared to their volume, which allows for efficient gas exchange directly with their usually aquatic surroundings. Their thin bodies also help. Now, moving slightly up the complexity ladder, we encounter organisms with, let's say, somewhat specialized structures. Take the earthworm, for example. It uses its moist outer skin, the cuticle, for respiration. This is called cutaneous respiration. The key word here is moist. Gases, especially O2, can only diffuse efficiently across a wet surface, so the earthworm's skin needs to stay damp. Insects have a fascinatingly different system, highly evolved, actually. They don't really rely on blood to transport the bulk of their gases. Instead, they use a remarkable network of finely branched tubes called tracheal tubes. These tubes transport atmospheric air directly within the body, reaching individual cells and tissues. It's like a direct air pipeline system. This reduces their reliance on the circulatory system for gas transport, making it incredibly efficient for their small size and often high metabolic rates. For aquatic arthropods, like crustaceans and many mollusks, living in water presents the challenge of extracting dissolved oxygen. They've developed specialized, highly vascularized structures called gills. This is branchial respiration. Gills are usually feathery or plate-like, with a huge network of blood vessels designed to maximize the surface area exposed to water for efficient O2 extraction. And then, for most terrestrial forms, including the majority of vertebrates like us, the evolutionary solution is lungs. These are internal, vascularized, bag-like structures specifically designed for efficient gas exchange with air. This is pulmonary respiration. If we look specifically at the vertebrates, we see further interesting specializations. Fishes, being entirely aquatic, they exclusively use gills. Amphibians, like frogs, show amazing adaptability. They do have rudimentary lungs for breathing air, but they can also respire through their moist skin. That's cutaneous respiration again, especially when underwater or hibernating. Some even use their mouth lining. This dual capability lets them thrive in both water and on land. Reptiles, birds, and mammals, being primarily terrestrial, they predominantly respire through well-developed lungs. What's truly fascinating here, as you see, is how evolution has sculpted these very specific, highly efficient solutions for gas exchange based on the environmental demands and metabolic needs of each organism. No single way works for everyone.
That's an incredible overview covering so much diversity. Now let's shift our focus back to ourselves, to humans. Could you please walk me through the human respiratory system? Step by step, explaining the pathway air takes from where it enters right down to where the actual exchange happens. I really need to visualize this clearly for my biology exams.
Absolutely. The human respiratory system is, well, a marvel of biological engineering. It's exquisitely designed for maximum efficiency in gas exchange. Let's trace the path step by step. Pay close attention to the structures and their functions. These are very frequently tested in your competitive exams. First, air enters your body through a pair of external nostrils. These are the openings located just above your upper lips. Pretty straightforward. From the nostrils, the air moves into the nasal chamber via the nasal passage. Now, this chamber isn't just a simple pipe. It's lined with fine hairs and a mucous membrane. These act as a crucial first line of defense. The hairs filter out larger dust particles, while the mucus traps smaller particles, humidifies the incoming air, and also starts warming it up towards body temperature. This conditioning role is important. The nasal chamber then opens into the pharynx. This is a critical anatomical junction. Why? Because the pharynx serves as a common passage for both food heading to the esophagus and air heading to the larynx. This dual role is a classic exam question point, so remember the pharynx is common to both systems. From the pharynx, the air proceeds into the larynx region. The larynx is often called the sound box. It's a cartilaginous structure made of various cartilages like the thyroid cartilage, the Adam's apple in males. Its primary job is sound production via the vocal cords. Now, a crucial detail here, very common in exams, is the epiglottis. This is a thin, elastic, cartilaginous flap, its vital function. During swallowing, the epiglottis automatically flips down to cover the glottis, which is the opening of the larynx, into the trachea. This clever mechanism prevents food from going down the wrong pipe, the trachea, ensuring it goes correctly into the esophagus. It prevents choking. Next in line is the trachea, commonly known as the windpipe. It's a straight tube extending down through your neck into the mid-thoracic cavity, or chest cavity. The trachea doesn't stay as one tube, it branches, specifically at the level of the fifth thoracic vertebra. That's an important anatomical landmark. Here it divides into a right and a left primary bronchi, deciacal bronchus. Each primary bronchus then enters a lung and starts dividing repeatedly, becoming narrower and narrower. They branch into secondary bronchi, then tertiary bronchi, and these keep dividing further, eventually leading to a network of very thin tubes called terminal bronchioles. This whole branching structure is often called the bronchial tree. Now, a key structural feature of the trachea, the primary, secondary, and tertiary bronchi, and even the initial bronchioles, is that they are supported by incomplete cartilaginous rings, C-shaped rings, essentially. These rings are vital. They prevent the airways from collapsing during breathing, keeping them open for air passage. Why incomplete? Why C-shaped? Open at the back. That's a clever design. It allows the esophagus, lying right behind the trachea, to expand slightly when you swallow food. Finally, each terminal bronchiole gives rise to a number of very thin-walled, irregular-shaped, and highly vascularized, meaning rich in blood capillaries, bag-like structures called alveoli, singular alveolus. These millions of tiny alveoli are the true microscopic workhorses. They are the primary sites where the actual gas exchange takes place between the air and the blood. This entire complex branching network, bronchi, bronchioles, and all the alveoli collectively makes up your lungs. You have two lungs situated on either side of your thoracic cavity. Each lung is enclosed by a double-layered protective membrane called the pleura. Between these two pleural layers, there's a thin film of pleural fluid. This fluid acts as a lubricant. It significantly reduces friction as the lungs expand and contract during breathing, allowing them to slide smoothly against the chest wall. The outer pleural membrane is attached to the thoracic lining, while the inner pleural membrane sticks closely to the lung surface itself. Functionally, it's helpful to divide the respiratory system into two main parts. This distinction is important for understanding its roles. First, the conducting part. This includes everything from the external nostrils down to the terminal bronchioles. Its main jobs are to transport the air to the alveoli, to clear it of foreign particles using mucus and cilia, to humidify it, and to bring it to body temperature. It's basically preparing the air. Second, the respiratory or exchange part. This consists only of the alveoli and their thin ducts. This is where the action happens, the actual diffusion of O2 and CO2 between the air in the lungs and the blood in the capillaries. Lastly, understanding the thoracic chamber is vital. The lungs are situated within this chamber, which is anatomically an airtight compartment. It's structured like this. Dorsally, back by the vertebral column, ventrally, front by the sternum breastbone, laterally, sides by the ribs, and on the lower side by the large dome-shaped diaphragm muscle. This anatomical setup is critical for breathing mechanics. Why? Because any change in the volume of this airtight thoracic cavity will directly cause a similar change in the lung or pulmonary volume. The lungs are passively linked to the chest wall. This is essential because we can't directly expand or contract our lungs by themselves. We change the thoracic volume using muscles, and the lungs follow along.
Wow, that's a very thorough breakdown of all the structures. Extremely helpful. Now that we know the anatomy, how does all of this actually work to get air in and out? You said we don't actively pump it. So what's the fundamental mechanism of breathing? How does it happen?
You're absolutely right. We don't pump it like a bicycle pump. The entire movement of air, both in and out, is driven by a simple but elegant physics principle, creating a pressure gradient. Specifically, a pressure difference between the air inside the lungs called the intrapulmonary pressure and the atmospheric pressure outside our body. This is a core concept you must grasp. Air always moves naturally from a region of higher pressure to a region of lower pressure. That's the key. Breathing involves two main stages: inspiration, inhaling air in, and expiration, exhaling air out. Let's look at each one. First, inspiration. This is drawing atmospheric air into the lungs. For this to happen, the pressure inside the lungs, the intrapulmonary pressure, must become less than the atmospheric pressure. We need to create a negative pressure, essentially. How do we achieve this? It's initiated mainly by the contraction of two sets of muscles. Number one, the diaphragm. This big dome-shaped muscle at the base of the chest cavity contracts. When it contracts, it flattens and moves downwards. This increases the volume of the thoracic chamber in the front to back direction, the anterior-posterior axis. Number two, The external intercostal muscles. These are muscles found between your ribs. When they contract, they pull the ribs and the sternum upwards and outwards. This action increases the thoracic volume in the side-to-side and back-to-front directions, the dorsoventral axis. So the combined effect of the diaphragm contracting downwards and the ribs lifting up and out is a significant increase in the overall thoracic volume. Since the lungs are stuck to the inside of the thoracic wall via the pleura, this increase in thoracic volume directly leads to a similar increase in the pulmonary volume. The space inside the lungs expands. Now, the physics kicks in. Boyle's law tells us that if you increase the volume of a container holding gas, the pressure of the gas inside decreases. So this increase in lung volume decreases the intrapulmonary pressure, making it lower than the atmospheric pressure outside. Since the pressure inside is now lower, air from the atmosphere naturally rushes into the lungs until the pressures equalize. That's inspiration. Think of a syringe. Pulling the plunger out increases the volume, decreases the pressure inside, and draws fluid in. Same principle.
Okay, contraction increases volume, which decreases pressure, pulling air in. Got it. What about getting the air out then? Expiration.
Right, now for expiration. This is releasing the alveolar air out from the lungs. For expiration to happen, the opposite condition is needed. The intrapulmonary pressure must become higher than the atmospheric pressure. We need a positive pressure gradient to push air out. Under normal, quiet breathing conditions, expiration is largely a passive process. It doesn't require active muscle contraction. It's achieved mainly through the relaxation of the muscles that contracted during inspiration. The diaphragm relaxes and moves back up to its dome shape. The external intercostal muscles relax, allowing the ribs and sternum to move back down and inwards due to gravity and elastic recoil. This relaxation reduces the thoracic volume. And because the lungs are elastic, they naturally recoil inwards as well, reducing the pulmonary volume. As the volume inside the lungs decreases, the intrapulmonary pressure increases to slightly above atmospheric pressure. This positive pressure gradient now forces the air out of the lungs until the pressures equalize again. Back to the syringe analogy. Pushing the plunger in decreases the volume, increases the pressure, and expels the fluid. It's important to note, though, that while normal expiration is passive, we can force air out more strongly. This is forced expiration. We achieve this by actively contracting other muscles, particularly the muscles in our abdomen and the internal intercostal muscles. These help to compress the thoracic cavity even further. We use forced breathing, both inspiration and expiration, during things like heavy exercise or when we need to clear our airways, like coughing or sneezing. On average, a healthy adult breathes about 12 to 16 times per minute at rest. This is a key value often asked in exams, so make a note of it. And we can actually measure the volumes of air involved in these breathing movements using a device called a spirometer. This instrument is really important clinically for assessing pulmonary functions, helping doctors diagnose lung problems. Understanding spirometry is directly linked to understanding how well someone's lungs are working.
That's a very clear explanation of the mechanics. Pressure changes, muscle actions. It all makes sense now. Now, you mentioned measuring air volumes. The NCERT textbook talks about respiratory volumes and capacities. Are these just different ways to measure how much air we move? And why are they clinically important, like you said, with the spirometer?
Exactly. These respiratory volumes and capacities are precise quantitative measurements. They help us evaluate how well the lungs are functioning. They are absolutely crucial for clinical diagnosis, for tracking respiratory diseases over time, and just for assessing overall respiratory health. For your exams, especially NEET, you definitely need to memorize these definitions and their approximate average values. They quantify the efficiency and capability of the lungs. Let's break down the basic measurements first, the fundamental respiratory volumes. 1, tidal volume TV. This is the amount of air you inspire or expire during a normal, quiet breath, just your regular, effortless breathing. The approximate average value for TV is about 500 milliliters. So if a healthy person takes, say, 12 to 16 breaths per minute, they move about 6,000 to 8,000 milliliters. That's 6 to 8 liters of air per minute. That's just TV multiplied by the respiratory rate. This calculation might pop up in objective questions. 2, inspiratory reserve volume, IRV. This is the extra amount of air you can forcibly inhale after a normal tidal inspiration. Take a normal breath in, then try to suck in as much more air as you possibly can. That extra volume is your IRV. It's quite large, averaging 2,500 milliliters to 3,000 milliliters. It represents your reserve capacity for deep inhalation. 3, expiratory reserve volume, ERV. This is the additional volume of air you can forcibly exhale after a normal tidal expiration. Breathe out normally, then push out as much extra air as you can. That's ERV. This typically averages around 1,000 milliliters to 1,100 milliliters. 4, residual volume, RV. This is the volume of air that always remains in your lungs, even after you've forced out as much air as possible after maximal expiration. You can never completely empty your lungs. The approximate value for RV is 1100 milliliter to 1200 milliliter. This residual air is super important. It prevents the tiny alveoli from collapsing completely, which would make it very hard to reinflate them. It also ensures that gas exchange can continue between breaths, smoothing out the oxygen and CO2 levels in the blood.
Okay, so those are the basic building blocks, TV, IRV, ERV, and RV. What about the capacities then? Are they just combinations?
Precisely. The pulmonary capacities are derived by adding two or more of the basic volumes together. They give a broader picture of lung function and are very commonly used in clinical settings. 1. Inspiratory capacity, IC. This is the total amount of air you can inhale starting from the end of a normal, quiet expiration. So it includes your normal breath, in plus your extra inspiratory effort. Formula, IC equals TV plus IRV. 2. Expiratory capacity, SC. This is the total amount of air you can exhale starting from the end of a normal inspiration. It's your normal breath out plus the extra you can force out. Formula, EC equals TV plus ERV. 3. Functional residual capacity, FRC. This is the volume of air remaining in the lungs after a normal quiet expiration. It's the air left in your lungs at the resting end expiratory level. Formula, FRC equals ERV plus RV. This represents the reservoir of air in the lungs available for gas exchange between breaths. 4. Vital capacity, VPC. This is a very important one clinically and frequently asked about. It's the maximum volume of air a person can exhale after a maximal inhalation, or conversely, the maximum air inhaled after a maximal exhalation. It represents the total usable volume of your lungs for ventilation. Formula, VC equals ERV plus TV plus IRV. It's a key indicator of lung health, often reduced in restrictive or obstructive lung diseases. 5. Total lung capacity, TLC. This is the absolute maximum volume of air your lungs can hold after the biggest possible inhalation. Formula, TLC, PLV plus ERV plus TV plus IRV. Or more simply, TLC plus vital capacity, VC, plus residual volume, RV. Understanding these definitions of formulas and the approximate values is crucial. It's not just theory. It's used daily in diagnosing and managing respiratory conditions. So definitely commit these to memory for your exams.
Right. Volumes are the basic measures, capacities are combinations. Got it. So we've covered how air moves and how we measure it. But now, the crucial part. Where and how exactly do oxygen and carbon dioxide swap places between the air and the alveoli, the blood, and our body tissues? Is it like an active pumping process or something simpler?
It's actually elegantly simple in principle, though highly efficient. It's not active pumping. It's driven by simple diffusion. The primary sites for this gas exchange are, as we mentioned, the millions of tiny alveoli in the lungs. But remember, exchange also occurs between the blood and the body tissues throughout your system. The key mechanism, both in the lungs and tissues, is simple diffusion. What does that mean? Gases passively move from an area where their concentration, or more accurately, their partial pressure, is higher to an area where it's lower. This pressure gradient is the main driving force. Now, other factors also influence how fast this diffusion happens. Two important ones are the solubility of the gases in the fluid, like blood plasma, and the thickness of the membranes the gases have to cross. Interestingly, carbon dioxide is about 20-25 times more soluble in water and plasma than oxygen. This high solubility helps CO2 diffuse efficiently even when the pressure gradient isn't huge. And the diffusion membrane in the lungs is incredibly thin, which also speeds things up. To really understand the driving force, we need to talk about partial pressure. In a mix of gases, like air, each gas exerts its own pressure independent of the others. That individual pressure is its partial pressure. We write it as PO2 for oxygen, PCO2 for carbon dioxide. Understanding these specific values across different locations is critical for NEET. Let's walk through them step by step using the typical values in millimeters of mercury, millimeter Hg. 1. Atmospheric air, what you breathe in, PO2 is high, around 159 millimeter Hg, PCO2 is very low, about 0.3 millimeter Hg. 2. Alveoli, air sacs in your lungs, a PO2 drops to about 104 millimeter Hg, PCO2 rises to about 40 millimeter Hg.
Wait, why does the PO2 drop from 159 to 104 in the alveoli?
Good question. It's because the fresh air you inhale immediately mixes with the stale air already in the lungs, the residual volume, which has less O2 and more CO2. Plus, CO2 is constantly diffusing from the blood into the alveoli, so the final mix has a lower PO2 and higher PCO2 than the outside air. 3. Deoxygenated blood arriving at the lungs from tissues. PO2 is low, around 40 millimeter Hg. PCO2 is high, around 45 millimeter Hg. This blood has given up its O2 to the tissues and picked up CO2 waste. 4. Oxygenated blood, leaving the lungs, going to tissues. PO2 rises significantly to about 95 millimeter Hg. Note, not quite 104 due to some physiological shunting, but close. PCO2 drops back down to about 40 millimeter Hg. This blood is now rich in oxygen, ready for delivery. 5. Tissues, active body cells, PO2 is low again. Around 40 millimeter HG because cells are constantly using O2. PCO2 is high. Around 45 millimeter HG because cells are constantly producing CO2. Now look at the gradients for diffusion created by these differences. For oxygen, O2. Lungs, alveoli, PO2 104, 90 keep deoxygenated blood, PO2 40. A big gradient, 104/40, excesses 64 millimeter Hg, strongly pushes O2 into the blood. This is oxygen tissues. Oxygenated blood, PO2 95, negative tissues, PO2 40. Another strong gradient, 95/40 equals 55 millimeter Hg, pushes O2 out of the blood and into the cells. This is oxygen unloading. For carbon dioxide, CO2, tissues, PCO2 45 millimeter H blood, PCO2 40 in arriving arterial blood. Gradient 45/40 equals 5 millimeter Hg, pushes CO2 into the blood. This is CO2 loading. Lungs, deoxygenated blood, PCO2 45 millimeter Hg, pushes CO2 out of the blood into the alveolar air to be exhaled. This is CO2 unloading.
The CO2 gradients seem much smaller than the O2 gradients.
They are, but remember CO2's high solubility. That compensates, allowing very efficient CO2 exchange even with these smaller pressure differences. It's quite fascinating how these subtle gradients drive the whole process. And finally, the diffusion membrane itself at the alveoli. It's incredibly thin, designed for speed. It's made of essentially 3 layers. 1, the very thin single-cell layer of the alveolar wall, squamous epithelium. 2, the single-cell layer of the capillary wall surrounding the alveolus, endothelium. 3, a very thin basement membrane or substance sandwiched between these 2 epithelial layers. The total thickness is tiny, much less than a millimeter, maybe only 0.2 micrometers. So you have favorable gradients, high CO2 solubility, and this ultra-thin membrane. All these factors work together perfectly for rapid, efficient diffusion of O2 into the blood and CO2 out of the blood in the lungs and the reverse in the tissues. It's a beautifully optimized system.
Okay. Diffusion driven by partial pressure gradients across the super thin membrane. Makes sense. So once the gases have swapped places, how does our blood actually carry them around? I know hemoglobin is super important for oxygen, but what are the different ways O2 and CO2 travel in the bloodstream?
Excellent question. Once diffusion happens, the blood takes over as the transport medium. It's the body's delivery system. Blood is indeed the primary way O2 and CO2 get moved around. Let's first look carefully at the transport of oxygen. The vast majority, about 97% of the oxygen, is transported bound to hemoglobin, Hb, which is located inside your red blood cells, RBCs. Only a tiny fraction, about 3%, is carried simply dissolved in the blood plasma, the liquid part of blood. Now, hemoglobin, it's that famous red-colored iron-containing protein in your RBCs. Oxygen binds with hemoglobin in a reversible way. This is key. It forms oxyhemoglobin, HbO2. Reversible means it can bind O2 easily in the lungs, where O2 is high, and release it easily in the tissues, where O2 is low. Each molecule of hemoglobin is quite amazing. It can carry a maximum of 4 molecules of O2. What determines how much oxygen binds to hemoglobin? The main factor is the partial pressure of O2, PO2. Higher PO2 means more binding, higher saturation, but other things can influence this binding affinity. Important factors include the partial pressure of CO2, PCO2, the hydrogen ion concentration, H+, which reflects acidity or pH and temperature. These factors act like modulators. This relationship is beautifully shown by the oxygen dissociation curve. If you plot the percentage saturation of hemoglobin with O2 against the PO2, you get a characteristic sigmoid or S-shaped curve. This curve is incredibly useful. It helps us visualize how factors like PCO2, H plus pH, and temperature affect hemoglobin's grip on oxygen. Understanding shifts in this curve, right shifts meaning decreased affinity, left shifts meaning increased affinity, is vital for exams. Let's see how this works in the body. In the lungs, alveoli loading O2. Here, conditions strongly favor oxygen binding to hemoglobin. You have high PO2, 104 mmHg, low PCO2, 40 mmHg, lower H plus concentration, higher pH, and relatively lower temperature. All these factors increase hemoglobin's affinity for O2, allowing efficient loading. The curve shifts left here. In the tissues unloading O2, here the situation is reversed, favoring oxygen release. You have low PO2, 40 mmHg, high PCO2, 45 mmHg from metabolism, higher H plus concentration, lower pH, more acidic due to CO2 and lactic acid, and higher temperature from metabolic activity. These conditions decrease hemoglobin's affinity for O2, causing it to release oxygen where it's needed most. The curve shifts right here. This beautifully illustrates how hemoglobin acts like a smart delivery truck, loading up efficiently in the supply depot and unloading effectively at the destination tissues. Quantitatively, remember this value. Every 100 milliliter of oxygenated blood delivers about 5 milliliters of O2 to the tissues under normal physiological conditions, a key fact for NEET.
Wow, that S-shaped curve and the shifts make so much sense for targeted delivery. Okay, what about CO2 transport? Is it similar or more complicated?
CO2 transport is a bit more varied, reflecting its different chemical properties and the large amounts we need to remove. It's transported in the blood in 3 main forms. 1, About 20–25% bound to hemoglobin. CO2 can also bind to hemoglobin, but it attaches to a different part than oxygen. It binds to the amino groups of the globin protein chains, forming carbaminohemoglobin, HbCO2. This binding is mainly influenced by PCO2. High PCO2, like in tissues, promotes binding. Low PCO2, like in lungs, promotes release. Interestingly, PO2 also affects this. High PO2 in lungs actually helps push CO2 off hemoglobin, part of the Haldane effect. 2. About 70% is bicarbonate ions, HCO3, in plasma. This is the major route, the most important way CO2 is transported, and it involves a crucial enzyme, carbonic anhydrase. This enzyme is found in very high concentrations inside RBCs and tiny amounts in plasma. It dramatically speeds up the reaction between CO2 and water. Here's the reaction, which is reversible. CO2 plus H2O carbonic anhydrase, H2CO3 carbonic acid, HCO3 bicarbonate ion, plus H plus hydrogen ion. Let's trace this. At the tissue level, high PCO2. CO2 diffuses from cells into the blood and enters RBCs. Inside the RBC, carbonic anhydrase rapidly converts CO2 and water into carbonic acid, H2CO3. This quickly breaks down into bicarbonate, HCO3, and hydrogen ions, H+. The bicarbonate ions, HCO3, then mostly move out of the RBC into the plasma, where they travel to the lungs. To maintain electrical balance, chloride ions, ClPl, often move into the RBC. This is called the chloride shift. The hydrogen ions, H+ produced, are largely buffered by hemoglobin inside the RBC, preventing drastic blood pH changes. At the lung level, low PCO2. The whole process reverses. Bicarbonate ions move back into the RBCs from the plasma. They combine with H+, released as hemoglobin binds O2. This reforms carbonic acid. Carbonic anhydrase then rapidly converts the carbonic acid back into CO2 and water. The CO2 diffuses out of the RBC, across the capillary and alveolar walls, and into the alveolar air to be exhaled.
So the carbonic anhydrase enzyme is key for this bicarbonate system to work quickly.
Absolutely critical. Without it, the reaction would be far too slow to handle the amount of CO2 we produce. This bicarbonate system is brilliant because it transports huge amounts of CO2 while also acting as a vital blood buffer, managing pH 3. About 7% dissolve directly in plasma. Just like oxygen, a small percentage of CO2 is simply carried dissolved in the plasma fluid. Because CO2 is more soluble than O2, this percentage is slightly higher. And another key value for exams, every 100 milliliters of deoxygenated blood delivers approximately 4 milliliters of CO2 to the alveoli. So you see, both gases have sophisticated, multi-pronged transport strategies.
Right, 97% O2 on HB, 70% ACO2 as bicarbonate. Those are big numbers to remember. It's incredible how precisely it all works. But how does our body actually know when to adjust breathing? Like breathe faster during exercise or slower during sleep? What controls this whole process, matching it to our body's needs?
That's the regulation part, and it's essential for maintaining balance or homeostasis. Humans have a significant ability to maintain and moderate their respiratory rhythm to perfectly suit the demands of the body tissues. This control is primarily handled by the neural system, specifically by specialized centers in the brainstem. Let's pinpoint the key players in the brain. 1. Respiratory Rhythm Center. This is the main boss, located in the medulla oblongata region of the brainstem. It's primarily responsible for generating the basic automatic rhythm of breathing, that cycle of inspiration and expiration. Think of it as the breathing pacemaker. 2. Pneumotaxic Center. This center is located higher up in the pons region of the brainstem. Its job is to moderate or fine-tune the signals coming from the rhythm center in the medulla. How? Signals from the pneumotaxic center can basically limit the duration of inspiration. By cutting inspiration short, it effectively increases the respiratory rate, often making breaths shallower and faster. It helps smooth out the breathing pattern. Now, besides these rhythm controllers, the system relies heavily on feedback about blood chemistry. 3. Chemosensitive area. This is a specialized area located right next to the rhythm center in the medulla. It is extremely sensitive to changes in the levels of carbon dioxide, CO2, and hydrogen ions, H+, in the cerebrospinal fluid bathing the brain. If CO2 levels rise, which also increases H+ levels, making things more acidic, this area gets strongly activated. It then signals the rhythm center to increase the rate and depth of breathing, the goal, to blow off the excess CO2 faster and bring the pH back to normal. 4. Peripheral chemoreceptors. We also have sensors located outside the brain, in major arteries, specifically in the aortic arch and the carotid arteries, at the point where they branch in the neck. These receptors also detect changes in blood CO2 and H+ levels. They are also sensitive to large drops in O2 levels, though less sensitive to O2 than to CO2H+ under normal conditions. When these receptors detect significant changes, they send nerve signals back to the rhythm center in the medulla, prompting adjustments to breathing. Now, here's a critical point. often misunderstood and important for exams. The role of oxygen in regulating respiratory rhythm is actually quite insignificant under normal physiological conditions. Our breathing is primarily driven by the need to regulate CO2 and H+ levels to maintain proper blood pH. The system is much more sensitive to slight increases in CO2 than it is to moderate decreases in O2. Significant O2 sensitivity only really kicks in at quite low oxygen levels, like at high altitudes. So if you connect this all together, you see a sophisticated control system. The medulla sets the basic rhythm, the pons fine-tunes it, and chemoreceptors constantly monitor CO2, H+R, and to a lesser extent O2 levels, providing feedback to adjust breathing rate and depth precisely to meet the body's metabolic demands and maintain that crucial internal balance, or homeostasis.
That's fascinating. So it's mostly driven by CO2 and pH, not O2 under normal conditions. Good to clarify that. Okay, this regulation sounds incredibly precise. But inevitably, sometimes things go wrong. What happens when the respiratory system gets disrupted? What are some common disorders students should be aware of for exams like NEET?
Absolutely. Understanding the disorders is a key part of the syllabus. Knowing what can go wrong helps appreciate how well the system normally works. For your exams, you should be familiar with a few common ones, focusing on the key features and causes. They often appear in match the following or definition questions. Let's start with asthma. This is quite common. The main symptom is difficulty in breathing, often causing a characteristic wheezing sound, especially when exhaling. The underlying cause is inflammation of the bronchi and bronchioles, the smaller airways. This inflammation makes the airways hyperresponsive, causing the smooth muscle around them to constrict, narrowing the passage and making airflow difficult. Allergens, infections, exercise, cold air can trigger attacks. Next, emphysema. This is a chronic disorder, meaning it develops slowly over time and is long-lasting. It's often grouped with chronic bronchitis under the umbrella term COPD, chronic obstructive pulmonary disease. The core problem in emphysema is damage to the alveolar walls. These delicate walls break down, leading to larger irregular air sacs instead of the tiny efficient clusters. This significantly decreases the respiratory surface area available for gas exchange. Lungs also lose elasticity. A major cause, and this is a direct link you must note, is cigarette smoking. The toxins in smoke cause chronic inflammation and damage. Finally, let's touch upon occupational respiratory disorders. These are caused by long-term exposure to harmful dusts or fumes in certain workplaces or industries. Think about grinding, stone breaking, mining, leading to halitosis or black lung disease, or working with asbestosis. In these situations, the body's natural defense mechanisms, like mucus and cilia, get overwhelmed by the sheer amount of inhaled dust. Long exposure leads to chronic inflammation, which can then cause fibrosis. Fibrosis is the scarring and thickening of lung tissue due to the proliferation of fibrous connective tissues. This makes the lungs stiff and causes serious, often irreversible, lung damage, impairing function severely. The key preventative measure here is for workers in such industries to consistently wear protective masks. This highlights the real-world health importance. Understanding these disorders helps us appreciate the fragility and complexity of this vital system, and underscores the importance of things like avoiding smoking and using protective gear in relevant occupations. So, to quickly recap our deep dive today on breathing and exchange of gases. We began with the fundamental need for O2 for energy and the removal of waste CO2. We journeyed through the diverse respiratory organs across the animal kingdom, seeing evolutionary adaptations. Then we meticulously dissected the human system, the conducting pathway preparing the air, and the exchange part, the alveoli, where the magic happens. We grasped the mechanism of breathing, driven by pressure gradients created by muscle action. We quantified lung function using volumes and capacities, remember those values. We then explored the core principle of gas exchange. Simple diffusion, driven by partial pressure gradients across that incredibly thin membrane. We followed the transport routes for O2, mostly hemoglobin, and CO2, mostly bicarbonate, thanks to carbonic anhydrase. And finally, we looked at the sophisticated neuroregulation, mainly driven by CO2 and H+A, and touched upon common disorders like asthma, emphysema, and occupational lung diseases. This chapter, yes, it's packed with details, concepts, values, but breaking it down step by step, as we've tried to do, makes it much more manageable. Make sure you revise those partial pressure numbers, the capacity formulas, and the functions of each part. Definitely practice past year questions on topics like the oxygen dissociation curve shifts and CO2 transport mechanisms for NEET. And remember, persistent revision and clarifying any doubts you have, that's your best strategy for success in these competitive exams.
That was incredibly detailed and so clear. Honestly, I feel much more confident about tackling this chapter now. especially with the exam focus and the way you broke down the really complex parts like transport and regulation. Thank you so much for this deep dive. It really was like a personalized tuition session. And to all of you listening and preparing, we genuinely hope this deep dive helps you master this essential topic for your exams. Keep digging for knowledge, stay focused, and we'll be back soon with another deep dive into a fascinating biological subject. And maybe something to think about. How might extreme environments, say very high altitudes with thin air or deep sea diving under immense pressure, challenge our respiratory system's ability to maintain these delicate gas exchanges? What kind of physiological adaptations might be needed to cope in those situations? Something to ponder.